Testing. Okay, so already 8 o'clock. Okay, so we will start our class now. So I see my line is okay or not. Bear with me. I think my line is okay. So hope everyone can hear my voice and can see the screen right. Okay, so actually for today we will learn about continue chapter 17, inheritance and genetic control. And so for today we will proceed to DNA replication topic. Okay, so actually I think these terms DNA replication, I think everyone is familiar to it, right? You already learned it in SPM, right? And from SPM until now. So actually DNA replication meaning, if you know that what is meaning of replication, meaning that DNA make a copy. So as you guys know, our cells, our molecule actually can DNA has have their lifespan, right? So actually our DNA cannot survive until we are in baby stage, until we are die. So cannot. So actually our DNA can be make a replication own self. So this topic you will learn about the DNA replication mechanism. So these are the topic that we will learn. So actually we will not only learn the mechanism of DNA replication, we will also learn the history of the DNA replication. So they need to learn the history, learn all the experiments. So there are total 3 experiments you have to learn. You have to learn to know why you want to learn this experiment. You need to learn about the mechanism, why this how these scientists can found the DNA replication mechanism. So firstly, this experiment, Fred Griffith experiment, is about to prove that DNA is act as the genetic material. It carry genetic material one. So these 3 experiment is to prove that DNA is genetic material. It carries genetic material. So actually, before the scientists not found that the DNA replication yet, actually the scientists even not know DNA is the molecule that carry genetic material. The scientists only know that nucleus carry genetic material only. Right. Our cells contain eukaryotic cells got nucleus, right? So they only know scientists at that time only know that uh the uh the nucleus carry genetic material. But which molecule? DNA, RNA, carbohydrate, protein, or others, they don't know. At that time, all the scientists don't know. So, how we can know that DNA carry genetic material? Who proved that? These 4 scientists, not five, 6 scientists, 3, 4, 5, 6. These 6 scientists, they do the 3 experiments and then prove that DNA carry genetic material. So what are the experiments you need to know is first experiment is the Fred Griffith experiment. So this is the scientist, he did the experiment at that time. So what does this experiment do? So if you want to know the experiment, firstly you need to know the objective. Why at that time he need to did the experiment like this, and then what are the procedures, what are the protocol, what are the steps it conduct the experiments, and also what are the result. So firstly you need to know is M, objective or objective. Next is your procedures. What are the experiment procedures? And then third one is result. You need to, if you do an experiment, you should have a result, right? And then finally, you do a conclusion. So what are the conclusions he made at that time? So if you see this experiment actually it conducted experiment is to actually the main aim is to test the it need to test the disease. One kind of disease we call as Streptococcus pneumoniae. This bacteria actually it contain either pathogenic or non-pathogenic. It will cause the disease one. And then when the pathogenic have the mouse have this pathogenic bacteria, it will directly die. Okay, so this experiment, the main aim is to actually to check the disease one. Want to check the disease. So there are two strains you need to know: S strain and also S cells and also R cells. So S is we call as smooth strain, and then R is about the rough strain. Okay, so S cell is actually is pathogenic, and then R cell is non-pathogenic. So if they did the experiment separately. So if the mouse is injected with the pathogenic control, which is pathogenic bacteria, then the mouse will directly die one. Dia akan mati punya. Okay, it will directly die. And then if the mouse is injected with the non-pathogenic cells, the mouse is remain healthy. It does not have any it will not die. Okay, however, if let's say we before let's say before injection, we kill the S cells. S cell is pathogenic, right? We know that S cell is pathogenic. Then we kill the S cells. Directly, we kill the S cell. So, meaning that for this injection, the S cell is already died. Already died already. Although it is pathogenic, but it already died. So it means that it also act as a non-pathogenic lah. So when we inject this kill S cells into the mouse will remain survive. It will not die. Okay, this is the third experiment. Third step, and then the final step is the most important and interesting step. That if let's say it it gives the kill S cells. S cells it already killed. It is already died already. It only have the dead X cells only. Maksudnya hanya mayat sahaja. Hanya mayat untuk X cells sahaja. Lepas tu, it campur add with the living R cells. So R cell actually you know it is non-pathogenic. So you add the living R cell into it. So meaning that this culture, this medium, this injection, it will contain living R cell and also dead S cells. So when these two are injected together with the mouse, they found that mouse is die. This scientist found that the mouse would die. And then when it get the blood sample, however it already died, the mouse is died, it did get the blood sample from this dead mouse, and it found that living S cells were found. And they could reproduce more S cells. So it is very interesting because at that time, at your beginning step, you only inject kill S cells, dead S cells. But finally, you found that in the mouse blood sample, you found living S cells. Kan dia sudah mati kan, S cell sudah mati, tapi tiba-tiba inject campur dengan living R cell, tiba-tiba dia macam hidup balik. It survive again. Dia hidup balik punya, macam zombie punya. Walaupun dia sudah mati, tapi dia pun boleh bergerak lagi. Dia macam zombie. So scientists found the interesting fact at here. So from here you can make one conclusion is the living R bacteria had been transformed into pathogenic S bacteria by an unknown. And it is the heritable substance from the dead S cell that enable R cell to make capsule. So meaning that the living R cell, so the key point is that the living R cell has been transformed into pathogenic S bacteria, meaning that R cell is already modified, changed to S cells already. Although it is R cell, at the beginning it is R cell, but got some substance, heritable substance from the dead S cells, enable R cell to become active S cells again. So it is very interesting because S cell, although it is die, but the genetic material, the information, all the biological molecule are still inside the cells. So although it is die, so all the biological molecule are inside the cell. So it found that got one certain biological molecule, normally is heritable substance. It can transfer into the R cells and make the R cells uh, become pathogenic. Actually, R cell is non-pathogenic, but it become the pathogenic and then become S cells again. So the scientists found this fact, but it also don't know which substance. It only know heritable substance only. But what substance, he don't know. At that time, he don't know. So it only know that got one substance that can heritable can be changed like that. So this experiment actually does not prove that DNA is genetic material lah. It only found that got one material is heritable. Dia hanya cari je. Tapi siapa tu, dia pun tak tahu. So this is the first experiment. And also this experiment, so you see that this experiment is was done at 1938. So it's very long period lah, already maybe 100, almost 100 years already from now. Okay, so they say the conclusion is it had the hereditary material had passed from that bacteria to the live bacteria that caused the harmless SR cell change to the S cell by transformation. So it only found that got one hereditary material, but what material he don't know also. At that time also he don't know. So how to prove it? So another experiments and another three scientists, Oswald Avery, Michelin McCarthy, and Colin McLeod. These three scientists prove it. It proved these three experiments. Not three experiments. Well, this experiment proved it DNA as the genetic material. Okay, so how they prove it? So they just modified the experiments from this Fred Griffith one. It also used the same cells as Streptococcus pneumoniae, Streptococcus pneumoniae. These cells also and also use mouse also. So as you see that it use only S strain lah. Dia guna S strain je. Okay, so it only use the pathogenic one because if the cells the mouse is injected with pathogenic strain, it will directly die by. Okay, so this experiment how it do it is it destroy the certain biological molecules. Meaning that when it injected when before injection it culture the bacteria and then for this one, it do the centrifugation all of that because it already found that got one heritable material, but what material he don't know. So these three scientists they use the reduction method lah, macam reduction method, one by one reduct. So firstly, they make a centrifugation, differential centrifugation that you learn in semester one. Semester one, break down all of the biological molecule, break down all the cell, release all the biological molecule, all the organelles, and then for here, it destroy all the polysaccharide. So all the carbohydrate, it destroy all. So meaning that this medium, this injection, it does not contain any polysaccharide, but others still remain. So when it is injected with the mouse, it found that the mouse still die. Mati juga. And then when it destroyed all the lipid, it hydrolyzed all the lipid material, it found that mouse also died. And then it destroyed all RNA, destroyed all RNA, others remain, it also die also. And then when it destroyed all the protein, others remain, it also found that mouse also died. But when it destroyed DNA, when DNA is destroyed, it found that mouse is survived. Mouse is survived. And then after they die, this one they die, they analyze their blood sample, they found that life exchange is detected, is recovered. But for this one, no life exchange at all. Tak ada punya. No at all. So meaning that this is the S train. It make a centrifugation, release all the components of the S train, and then it injected R strain. So this is the R cell. You see that this one is R cell. So because it already say that even though S train is die, but if it contains the heritable material, or when it injected combined with the R cells, because this heritable material can be transformed into R cell and then become S cells again. So for this experiment is to detect which material. So we cancel one by one, we do one by one. When we destroy polysaccharide, cells die. Destroy lipid, RNA, protein, all cells die. But when we destroy DNA, meaning the cells still survive. So it means that they made a conclusion that DNA is the heritable material that cause the cells that can transform into the R cells and then cause pathogenic and then make the cells die. Why? Because when we destroy DNA, the mouse is survived already. So this is the overall experiments. Okay, so this is the explanation. Okay, so it will give the strong evidence that DNA is one that carry the genetic information rather than other molecule. Okay, however, at that time, this I don't know which year, I forget to mention, but you can Google it which year. But however, at that time, many scientists got objection about this experiment result. Banyak researcher, scientist tu tidak percaya. They don't think that this experiment can 100% prove that DNA is carrying genetic material. They cannot accept that. Because why? At that time, the information of DNA was little, because at that time, you want to analyze DNA is very difficult, because at that time, the technology is not very expert like as now. And then some scientists also say that only bacteria will follow this as every experiment. After these three